So good afternoon, everyone. Thank you once again for joining us here on this virtual format for our March lunchtime expedition presentation. Just a couple quick announcements before we introduce today's speaker. The support for these programs have been made possible through Sage Creek Ranch and the Nancy-Carroll Draper Charitable Foundation. These supporters really and the sponsors may help make everything that we do possible. So we are extremely grateful to them, as well as to you, our audience, for your feedback and for your support for programs like the Lunchtime Expedition program. Several of you have reached out over email and suggested speakers, and we definitely encourage that. If you come across a name or research that you find incredibly fascinating, feel free to send those along to us. We'll definitely check out those speakers, those researchers, and hopefully incorporate several of them into the program. So we are recording these lectures and uploading them to our Draper YouTube channel. Complete presentations from previous years are now available for your viewing pleasure. If you'd like to be added to our email list, if you're coming through us today, through Facebook, through Twitter, or through the Center of the West website or another medium, and you'd like to be added to the listserv, just shoot me an email at Corey A. That email will be on the outro slide, corey@centerofthewest.org, and we will get you on the announcement, the listserv for future presentations. We also include links to our previous presentations in those outgoing emails, so another good reason to sign up in case you missed one from earlier. We are broadcasting today's presentation over Zoom webinar and Facebook Live, so feel free at any point during the presentation, as a question or a comment arises, drop that in the chat feature, drop it in the Q&A feature, and we'll make sure that those get relayed to our speaker at the end of the presentation. So that said, it is my pleasure to introduce our speaker today, Dr. Emily Schultz. I've had the opportunity to speak with Dr. Schultz's first year biology students, and now today I'm delighted to highlight her as our featured March speaker. So Dr. Schultz started her career in ecology at Scripps College, working with Dr. Diane Thompson on interactions between native and invasive plants in Southern California. After college, Dr. Schultz traveled the world as a research assistant before completing her PhD in ecology and evolutionary biology with Dr. Tom Miller at Rice University. After graduate school, she worked as a postdoctoral researcher with Dr. Margaret Evans at the Laboratory of Tree Ring Research at the University of Arizona. To date, Dr. Schultz has helped study the cascading effects of brown tree snakes brown tree snake introduction on Guam, the recovery of endemic plant population following invasive herbivore removal on the California Channel Islands, and the effects of climate change on pine distributions. Dr. Emily Schultz is currently an assistant professor of biology at Northwest College, where she teaches general biology, plant and fungal biology, and forest management. Without further ado, we'll turn it over to Dr. Schultz.
All right, thank you so much, Corey, for your introduction and for inviting me here to speak. I'm really excited to be here sharing my research with you. So thanks to all of you who are here to listen, especially when you could be outside enjoying the sun in Wyoming right now. So today I'm going to be sharing some research that I worked on as a graduate student on plant-insect interactions, and specifically the role that native insect herbivores might play in limiting the invasion of exotic plants. So before I get into the research itself, I want to start off by thanking my co-authors on this project. I actually came to this project fairly late in the game as the population modeling expert. So I want to make sure I highlight the work that my co-authors did to set up this really cool experiment. So most especially, I want to recognize Jim Ekberg. So the research I'll be talking about today actually grew out of Jim's graduate work. So he designed the experiment and set it up and did most of the data collection, and then handed me this really amazing data set to work with. Sergey Berg did some of the preliminary analysis on the data so that I could just get started on the fun stuff once I had the data set. Tom Miller was my graduate advisor, so he guided me through a lot of this work. And finally, Svacha Luda is really kind of the giant on whose shoulders the rest of us are standing. So Jim and Tom were both Svacha's graduate students. And in addition to being an expert on invasive species and plant-insect interactions, she really developed the system that I'll focus on today. And she mentored an incredible generation of plant ecologists who have in turn mentored me. So I actually have multiple connections to scientists that she mentored. So she played a big role, not in just this research, but in my career as a plant ecologist. So today I want to start by sharing the story of two different species, the brown tree snake and the bull thistle. These are both species that got relocated to a new land where they could attempt to establish new populations. And I'll start off with the story of the brown tree snake, which is really a classic invasion story, the sort we hear again and again about species that get introduced to a new ecosystem where they then rapidly spread and it leads to devastating effects on those ecosystems where they were introduced. And in fact, the story of the brown tree snake is probably one of the world's best known invasion stories, and one that I was fortunate enough to be involved in when I was pretty fresh out of college. So just as a quick additional acknowledgement here, I want to recognize Huldra Rogers, who was my boss in Guam, and she's been leading research on the brown tree snake invasion in Guam for more than a decade now. So she's contributed a lot about what we know about the effect of this brown tree snake on the forests in Guam. So the brown tree snake is native to Australia and some of the surrounding islands like Papua New Guinea and Indonesia. But it was accidentally introduced to Guam in the 1940s, likely either by stowing away in a ship's cargo or in the landing gear of a plane. And then upon arriving in Guam, it found an island that was devoid of snakes because there are no snakes that are native to Guam. So this presented the brown tree snake with an incredible opportunity. So here's an ecosystem that was completely naive to snakes. So there were very few organisms on the island that would prey on these snakes. The few that would were primarily invasive species themselves. So without any predation or minimal predation, the brown tree snake spread rapidly through the islands. And along the way, it encountered really a feast of birds spread out before it. And again, these were birds who had never before encountered snakes and had no defenses against predators like this snake. So the brown tree snake just ate its way through these species one by one until there were almost no birds left on the island. So of the 12 native bird species on Guam, 10 of them were driven to extinction in the wild on Guam, leaving only tiny isolated populations of the remaining bird species. And this loss was stark and very obvious. So if you visit some of the other Mariana Islands that are found near Guam, such as Saipan and Rota, you can see or really hear the contrast between the forest on those islands and the forest in Guam. So these other islands are ecologically similar to Guam, but they don't have established populations of the tree snake. So their bird communities are still intact. So if you go out and walk in the forest on Saipan and Rota, you can hear the bird calls all around you and the megapodes shuffling their way through the leaf litter. But on Guam, the forests are almost completely silent. And this silence is indicative of a far greater loss to these forests, which is the loss of the functions that the birds provide in these forests, including their roles as arthropod predators and as seed dispersers. So my boss Huldra, when she started this research, she hypothesized that without birds in the forest on Guam eating herbivorous insects, there would be an increase in the predation of these insects on plants. And this actually turned out not to be true, because as it turned out, the birds were also important predators of spiders. So without the bird populations, there was a dramatic increase in the spider populations in the forests. And these spiders are insect predators themselves, so they were able to control the insect populations. However, of course, this comes at the expense of the forest now being full of spiders instead. So the results of this research actually led to some rather dramatic headlines, such as this one, that essentially described Guam as being overrun by snakes and spiders. And while these are something of an exaggeration, I certainly wouldn't describe Guam as nightmarish. And in fact, I love working there. It is really noticeable that there are more spiders on Guam than on the other islands. And when I applied for the job in Guam, I was actually asked if I'm afraid of spiders. because that would have been a challenging thing for working in the forest there. Now, the second major role of birds in the forests is to act as seed dispersers. So many of the island's native trees are adapted for seed dispersal by birds. They have these fleshy fruits that will attract the birds to come and eat them and then carry their seeds throughout the forest. And this dispersal is really important for the trees because it moves the seeds away from the parent tree and is more likely that those seeds are going to land in locations that are favorable for germination. And for some of these species, the seeds also have higher germination rates if they first pass through the guts of the birds. So with few remaining birds on Guam, the seeds primarily fall right below the parent tree, where their survival is typically lower. And they're also unable to fill in those gaps in the forest that can be created by the frequent disturbances in these forests, like typhoons. And some of these bird dispersed seeds are also culturally and economically important to the indigenous Chamorro populations. And one example is these wild peppers, which make a delicious hot sauce. So the birds are threatening not only the forest and the species in those forests, but also the human populations that rely on those forest resources. So again, the brown tree snake presents us with one of these classic invasion stories. And these classic stories are important. So for one thing, they really make us care about these issues, especially when we're talking about the loss of really charismatic animals like the birds on Guam. So, who wouldn't want to save the Mariana fruit tubs with their beautiful plumage, or the megapodes with their giant feet? But even aside from this kind of emotional aspect, these classic stories give us knowledge that's important for conservation and for understanding ecology in general. So they show us the potential devastating effects of invasive species, such as the exclusion of native organisms, the spread of disease, the disruption of nutrient cycling through ecosystems, and shifts in disturbance regimes like fire regimes. And altogether, these things can ultimately lead to a loss of biodiversity, a loss of ecosystem function that even humans rely on, and huge economic costs as we try to make up for these lost resources and put effort into trying to remove these invasive species. And these stories also tell us about the conditions that can lead to invasion, which can help us predict and try to manage these invasions. So we can learn both about the characteristics of the invading organisms that make them good invaders, and the characteristics of the ecosystems that can make them vulnerable to invasion. So some of the things that we've learned about invasive species is that they're often very good dispersers, and they have high reproductive rates and a short time to maturity. Many of them are often generalists or strong competitors as well. And this allows these species to rapidly spread through an area and outcompete or eat or infect the native species. And vulnerable ecosystems are often naive to the invading organisms. So that's what we saw on Guam with these birds that had never been faced with a snake predator before. These ecosystems might also lack predators or pathogens that would attack the invading organism. And often they have a frequent disturbance, and especially unnatural disturbance that might promote invasion, because many of the characteristics that make species a good invader also make them very resistant to disturbance. And finally, although this last one is somewhat debated amongst ecologists, communities that lack diversity might also be more prone to invasion. So these stories are important, but on their own, these classic stories still leave gaps in our knowledge about invasion. So there's this other side to invasions, and these are the often forgotten and many times completely unnoticed stories of the failed invasions. So these are organisms that arrive in a new place, but are unable to establish, or at least they're unable to establish dominance in the new ecosystem. And it's one of these failed invasion stories that I'm going to focus on for the remainder of my talk. So specifically, I would like to tell the story of bull thistles in Nebraska. Now, some of you, if you had experience with bull thistle, might be a bit perplexed by my reference to bull thistle as a failed invasion. And that's fair, because bull thistle is arguably one of the most successful invasive thistles in the United States. So this map here shows the distribution of bull thistle, where the blue is showing states where the species is present and exotic. The teal is showing counties where the species is present and exotic. And the purple is showing counties where this species is considered a noxious or invasive weed. So we can see that in many states, bull thistle is classified as noxious or invasive, and the species is found in pretty much every state in the country. And in places where the species is invasive, it can do things like outcompete the native plants, and due to things like its spininess, it's unpalatable to most large herbivores. So it can reduce the forage quality for both native herbivores and for livestock. And again, because of its spininess, it's literally painful to try to get rid of this species. But if we look at Nebraska specifically, we see kind of an interesting thing. So although Nebraska is surrounded on multiple states where bull thistle is considered invasive, it's not invasive in Nebraska itself. And I have it on good authority from friends who are from Nebraska that, yeah, you see it here and there, but its abundance is typically low and it doesn't seem to cause problems in the ecosystems there the way it does in some of the surrounding states. So although it proved itself to be a capable invader in many parts of the United States, in Nebraska, the invasion of bull thistle failed, which gives us an opportunity to further our knowledge of invasions and understand not just the conditions that are favorable for invasion, but also the conditions that can lead to the failure of potentially invasive species. And this knowledge that we can learn from these failed invasions is just as important as what we can learn from the classic invasion stories. So it can help us make better predictions about where species might invade and where we should focus our management efforts. So consider this diagram here showing some predictions for the potential distribution of an introduced species. So in this example, the species was introduced at 5 different points in this region. And if we only had information on the successful invasions, which is the scenario shown on top, where we only have data on those 2 successful invasions, we might predict that the species is capable of invading the entire region. And we would spread our resources thin as we try to prevent its spread. However, if we also have information about the failed invasions, as shown in the bottom row, we can refine our predictions and identify areas where the species is unlikely to invade. And that can allow us to focus our management efforts on the areas that are at greatest risk. And one thing that's critical to remember about these failed invasions, which is easy to forget or not even know in the first place, especially when we're constantly hearing these classic invasion stories, is that we actually expect most invasions to fail. So these forgotten stories of failed invasions are really the most common invasion stories. And if you think about what a species has to face when it gets relocated to a new area, it starts to make sense that most of these invasions would fail. So first, it's usually a very small subset of a population that gets relocated. And sometimes that could be only a single individual. And we refer to this as the propagule pressure, the number of individuals that gets moved to this new place. So it can cause problems for this potentially invading species if the new population size is too small. So in some species, there might be mate limitation at small population sizes, and this could prevent the reproduction and ultimately the growth of these populations. And there can also be genetic constraints. So small populations usually lack genetic diversity, and that can be problematic, particularly if the species is confronted with new and variable environmental conditions that it has to adapt to. But let's say this new population that gets started is large enough to be viable. Well, then the next thing that the species is confronted with are the abiotic conditions in its new environment. The abiotic conditions being the non-living components of the environment, such as the climate and the soil type. And every species is going to have a set of environmental conditions that it can withstand. So if the abiotic conditions in this new area are not suitable for this non-native species, it will be unable to persist in that new area. But if it does manage to persist under those abiotic conditions, it's then met with biotic resistance, or resistance from the living components of the ecosystem. So this could include competition from similar species, or it could be predation or herbivory or parasitism. And just like native species might be naive to an invader can also be naive to the predators and pathogens that it might encounter in a new ecosystem. And finally, many species have mutualistic relationships with other species that can aid or are even required for their survival. So one example for plants is that they often form mutualistic relationships with fungi in the soil. So these fungi will interact with the roots of plants and essentially increase the surface area of those roots, allowing the plants to absorb nutrients better. And these mutualistic relationships are really common. So if a species gets moved to a new location without those mutualistic relationships, if those mutualists don't move along with them, that could ultimately prevent their survival in the new environment, especially if there aren't any native species that could potentially fill those roles. So only if a species can overcome all of these barriers will it be able to persist in the new environment and potentially become invasive. However, although we have a theoretical understanding of why invasions might fail, we actually know very little about the importance of these different factors in preventing invasion. In fact, this was the topic of this review by Zenny and Nunez in 2013, which really drew our attention to the importance of failed invasions, as well as highlighting our lack of understanding, because these failed invasions are wildly understudied compared to the less frequent successful invasions. So in this review, Zenny and Nunez looked at studies that reported failed invasions. And they were actually only able to find 76 studies that reported these failed invasions. And that's in comparison to probably thousands of studies that have been done on successful invasions. So they recorded the factors that were suggested or tested as being the cause of those failures reported in these studies. And most of these studies actually neither suggested nor tested any factors that might have led to those failed invasions. Abiotic and biotic resistance were the most commonly suggested factors for these failures, but they were rarely tested. And the other factors, the mutualist release, propagules pressure, and genetic constraints, were only tested a few times. So there's a lot we still don't know about the role of these factors and how important they are in preventing invasion. So we use this opportunity of the failed invasion of bull thistle in Nebraska to help fill in the gaps in our knowledge about why invasions might fail in some places. And specifically, we focused on the role of biotic resistance in the form of native insect herbivores to possibly explain the failure of this bull thistle invasion. Now, even though I'm talking about invasive species management here, at heart, I'm really a basic ecologist. So I'm really interested in developing an understanding of how ecological systems, particularly populations, work. But I often work at the interface of applied and basic ecology because of the way the two approaches can inform each other. And this project is no exception. So this question about the role of biotic control of an invasive thistle can actually inform our more general understanding of plant-insect interactions and population dynamics. While understanding plant-insect interactions can then help us better understand the role of insects in controlling invasive species. So in this research, we kind of tackled both sides of this. So we used this thistle system to ask the more basic question of what role insects play in the regulation of population dynamics. And in turn, this helped us assess the potential that insects have for controlling non-native plant populations. I want to explain a little bit more about what I mean by the role insects play in regulating plant population dynamics and why it's something we still don't really fully understand. So what I specifically mean is, does insect herbivory limit or reduce plant population abundance? And this is really what we care about if we're thinking about insects potentially controlling invasive species. So it's already very well established in the study of plant-insect interactions that insects can affect individual plants. And depending on the insect and what it is they do, they might reduce the growth of the plant, or feed on their seeds and limit reproduction, or even kill the entire plant. But what we still don't really know is even if insects can affect individual plants, do these individual level effects lead to population level effects like reduced plant population sizes? Now, this is kind of counterintuitive that insects could kill plants, but not ultimately affect their population size. But this is actually something that happened because insects are not the only forces that are at play in limiting plant populations. So it's very well established that plants can also be affected by the density of their own populations. And this is due to things like resource limitation. So plant population growth rates can decline and even become negative if the population density gets too high. And we refer to this as negative density dependence. And if we look at negative density dependence in graphical form, it looks something like this, where we have density on the x-axis here and the population growth rate, which is abbreviated as lambda, on the y-axis. So as the density of the population increases, the population growth rate is expected to decrease. And at low densities, the population growth rate is likely going to be above 1, which means that the population is growing. But at high densities, the growth rate can drop below 1, meaning that the population would decline. And it would decline until it reaches a lower population size that can be supported by the environment. So this is related to insect herbivory and the role that herbivory might play in limiting population sizes. Because if insects are only killing off the excess individuals that can't be supported by the environment anyway, they would not actually reduce the population size. So this can be a little tricky to wrap your head around. So I like to think of it kind of in terms of planting a garden. So when you plant a garden, you usually plant more seeds than you actually need in case some of those seeds just don't germinate. But if you do have good germination rates, as a result of planting all these seeds, you could end up with a very dense population of plants in your garden. And if you left all of these plants to grow, then they could become overcrowded and likely they wouldn't grow as well. And some of the plants might even die off as a result of this overcrowding. So usually what you do if you end up with this very dense garden is that you would thin out the plants. So you would remove some of those individuals, and that would allow the remaining plants to survive better. And they might also compensate for the loss of those other individuals that were thinned out by growing larger and producing more fruit, which could then lead to larger population sizes in the future. So insect herbivores can actually act in the same way in nature. So acting as natural thinning agents that remove only the excess individuals and leave healthier plant populations in their stead. And classic ecological theory actually tells us that this is what we should expect. But it hasn't been rigorously tested because most experience and models of insect herbivory and population dynamics have not accounted for this negative density dependence in plant populations. And furthermore, even if we know that under some circumstances, herbivory can limit plant populations, it might also depend on the particular environmental conditions. And these are conditions that could vary over time and space. Some examples of these conditions might be things like the vegetation type, or the distance to the edge of a patch of vegetation, or climatic gradients. So even if insects limit plant populations under some conditions, they might not under other conditions. So for both a basic ecological understanding of plant populations and for making good management decisions, it's important to know how universal the impacts of insects on plant populations actually are. So to inform our understanding of the role of biotic resistance in preventing invasions, we use this failed invasion of bull thistle to test for the effects of both herbivory and density on plant populations to determine if insect herbivores can limit the abundance of plant populations and whether we would expect this to be a universal outcome in our study region of Nebraska. So specifically, we asked first, how do herbivory and density influence bull thistle population growth rates? And second, how do individual vital rates contribute to the overall influence of herbivory on population growth? And by individual vital rates, I mean things like the survival rate and growth rate and reproduction rate of individual plants in the population. And finally, does the suppression of bull thistle invasion by native herbivores vary with environmental context, or is it a general result? So we tested these questions using an experimental approach in combination with a population model. So the experiment was set up, again by my co-authors, across 8 sites in eastern Nebraska. And they set up 14 plots at each site. 7 of those plots were treated with insecticide to exclude insect herbivores, and 7 of those plots were left untreated, which allowed us to determine the effects of the insect herbivory on the plants. And the plots were also seeded with 7 different densities of thistle seeds, ranging from 150 seeds per plot to 1,050 seeds per plot. So when we collected the data on these thistles and these different treatments, we didn't just count the thistles to estimate the population growth rate. We instead measured these individual vital rates of the thistle, including the survival rate of the plants, their annual growth rate, the probability that these plants flowered, and if they flowered, the number of seeds that they produced. And finally, the probability of germination of these seeds. And with information on all of these vital rates, we could then put it into a population model that would allow us to calculate the population growth rate of the thistles. So this approach of measuring the vital rates instead of just population counts has several benefits. So the main reason is that not all vital rates respond to environmental factors, such as insect herbivory, in the same way. So in this example figure shown here, we have some environmental variable on the x-axis, and this environmental variable has a positive effect on the survival of the plant, but a negative effect on the growth. And this is something that we actually see very commonly when we look at populations. So by measuring the individual vital rates, we can determine the different ways these vital rates respond and how that ultimately affects the population growth rate of the population we're interested in. And this can be really important for helping us understand the system and helping us focus our management efforts, because we might want to focus our efforts on particular vital rates that matter most to the changes that we're seeing in the population growth rate. And for this study in particular, this approach of measuring individual vital rates is also useful for this question of whether insects limit populations, because it can help us determine if insects are acting as these natural thinning agents, which would then allow the remaining plants to compensate for decreased survival by increasing their growth and reproduction. So if populations are compensating for lower survival in this way, we would expect that herbivory might have a negative effect on the survival of the plants, but a positive effect on other vital rates like the growth and reproduction of the plants. So we can detect these differences by measuring these individual vital rates. And remember, under this thinning model, the insects would affect individual plants, but not the plant population abundance. So putting these data all together from the different vital rates to calculate the population growth rate can help us determine the effects of herbivory and density on the population growth rates of these plants. And because this experiment was replicated across 8 widely dispersed sites across eastern Nebraska, we were also able to estimate how these effects and ultimately the population dynamics of the thistle varied across space. And this is what allowed us to determine how universal the impacts of insects on the thistle are in Nebraska. Okay, so now that I've gone through our approach to answering these questions, I'll walk you through the results that we got, starting with the first question: how do herbivory and density influence bull thistle population growth rates? Now, I do also have data on the effect of herbivory and density on the individual vital rates. For the sake of time, I'm not going to show that to you, but if you are interested in seeing some of those effects, I'm happy to share them with you at the end if you just ask. But I do want to just briefly summarize those effects before I get into the population growth rates. So first, when we looked at the effect of herbivory on these vital rates, we found that herbivory had a negative effect on every single vital rate that we measured. So there was no sign of this compensation in response to herbivory that would support that natural thinning hypothesis, herbivory which is bad for these plants across the board. And when we looked at the effect of density, we found that density had a negative effect on growth, but a positive effect on the survival and probability of flowering of these thistles. So we then put all these vital rates together and made our population model and looked at the effects of density and herbivory on the population growth rate. So this figure here has the density of the plants on the x-axis and the population growth rate lambda on the y-axis. So remember, if the population growth rate is above 1, that indicates the population is growing. And if it's below 1, that indicates that the population is declining. So this dotted line here on the figure is showing where lambda is equal to 1. The solid line is showing the population growth rate under herbivore exclusion. So these were the insecticide treatments where the herbivores didn't have access to the plants. And then the dashed line is showing the population growth rate under herbivore access. So the first thing that's really obvious here is that in the absence of herbivory, these thistle populations would be able to grow very rapidly in Nebraska. So down here at these low densities, we're looking at population growth rates around 5, which means that in the next year, we would expect the population to be 5 times the size that it is in the current year. So without insects, we would expect very rapid growth of thistle populations. However, under herbivore access, the thistle populations declined. So the population growth rate was below 1, regardless of the density in the presence of these insect herbivores. So in other words, without insect herbivory, the thistle would be expected to invade. But in the presence of herbivory, the populations were unable to even establish. So then regarding the density and the effect the density had on the growth rate, we found that under herbivore exclusion, density had a negative effect on the growth rate. So eventually we would expect to see that these populations would decline when they got to very high densities, but higher than what we actually observed in this study. However, in the presence of herbivores, there was a slightly positive effect of density, but we didn't see a positive population growth even at the highest densities that we observed in this experiment. So to answer this first question, how do herbivory and density influence bull thistle population growth rates? We found that herbivory had a negative effect on bull thistle, driving those populations to decline. And density had a negative effect on bull thistle under insect exclusion, but a very slight positive effect under herbivore access. So now on to the second question, how do those individual vital rates that we measured contribute to the overall influence of herbivory on population growth? So in this analysis, what we were looking at was the difference in the growth rate between these two herbivory treatments. So we looked at how much lower the growth rate was under herbivore access relative to herbivore exclusion. And we calculated this across all of the densities we observed in the experiment. And what we'll look at is how these different vital rates contribute to the difference in the growth rate between our herbivory treatments. So this figure here, again, has plant density on the x-axis, and now it has the change in the population growth rate on the y-axis. So this is the difference in the population growth rate between herbivore access and herbivore exclusion. So this solid line that's being plotted here is the total difference in the growth rate between those two treatments. And it's negative because herbivory had a negative effect on the population growth rate. So now I'm going to add the additional lines showing the contributions of these different vital rates to this effect of herbivory that we saw. So these two lines down here at the bottom are for growth and seed production. So this tells us that growth and seed production contributed the most to the decline that we saw in the population growth rate between these two treatments. Recruitment, which is this dashed line here at the top, had a negligible contribution. And then survival and probability of flowering had intermediate contributions to this difference we saw. So altogether, this tells us that the decrease in the population growth rate as a result of the insect herbivory was driven primarily by the lower growth rates of individual plants and the decline in seed production in the presence of herbivores. So then finally, our last question was, does the suppression of bull thistle invasion by native herbivores vary with environmental context, or is it a general result? So to look at this, we first looked at the population dynamics we would expect at different locations in eastern Nebraska under herbivore exclusion. So this figure here is showing the different types of dynamics predicted by our model and how frequently we would expect to see those different dynamics across eastern Nebraska. And first of all, we found that we should expect a wide variety of dynamics in the absence of insect herbivores. And the most common one we would expect to see are these aperiodic fluctuations, which are characterized by very rapid growth of the population, followed by a very rapid decline of the population. And these sorts of dynamics are really common in populations that have very high growth rates and strong negative density dependence, like we saw in this thistle. And populations that fluctuate like that are also very difficult to predict and very difficult to control. The second most common dynamics we would expect to see in Nebraska are unbounded growth. So essentially the populations would just continue to grow and grow. Of course, they would have to stop growing eventually, but we didn't see high enough densities in our study to be able to estimate exactly where these populations would level off. We just know that they would get to very large population sizes before that happens. Then less frequently, we would expect to see things like stable oscillations or population decline. So there are some places where these thistles wouldn't be able to establish even without insect herbivores. And then least frequently, we would expect to see logistic growth where the populations would increase up to a point and then level off. Now, despite this wide variety of dynamics that we would predict under herbivore exclusion, the insect herbivores were able to suppress these thistle populations under almost all conditions. So this figure here is showing the frequency of growth rates that we would predict to see across Nebraska, both in the presence and the absence of herbivores. So we have the growth rate now on the x-axis and the frequency with which we would expect to see that on the y-axis. So under herbivore exclusion, which is shown here in dark gray, we predicted a wide range of growth rates, mostly very high, indicating that the thistles would typically invade, but there are a few below 1 where those populations would decline. So this is very much in line with what we saw in the previous figure, that most of these populations would be able to persist and usually with very high growth rates. But in the presence of herbivores, and these values are shown in the light gray bars, we predicted that 97% of the population growth rates across Nebraska would be below 1. So in almost every case, So to answer this final question, does the suppression of bull thistle invasion by native herbivores vary with environmental context? Our answer to that was no. So regardless of the underlying thistle population dynamics and environmental conditions, the insect herbivores were able to suppress bull thistle invasion. So to conclude, I just want to return to these big questions that I presented at the start. First, the basic question, do insects play a role in the regulation of plant population dynamics? And the applied question, how much potential do insects have for controlling non-native plant populations? So to the first question, does herbivory limit plant population abundance? The answer to this, based on our work, was yes. At least in this system, native insect herbivores not only limited the abundance, but drove them to decline, so they were unable to establish. And this was true despite the strong negative density dependence which we saw under herbivore exclusion, which could have set up the insects to act as those natural thinning agents. But the negative effects of herbivory were so strong in the system that they overwhelmed that negative density dependence and were able to affect the population abundance. And furthermore, the suppression was a general result across the study region. So regardless of the different environmental conditions across the study region, the insects drove the thistle populations to decline. Now, of course, this was just a single species that we were working with. So additional research on other species and particularly other studies of these failed invasions would be really important to see if this can also be generalized to other systems. However, the fact that insects are able to suppress these populations tells us that native insect herbivores do have strong potential for controlling or even preventing the establishment of potentially invasive plants, despite classic theory telling us that insect herbivores shouldn't affect plant population abundance. And I think based on this, we can kind of accept that this population of native insects provides a likely explanation for why bull thistle has been unable to invade in Nebraska. But what makes Nebraska so different from the surrounding states? So why don't the native insects in Colorado or South Dakota prevent bull thistle from invading in those states? Well, this wasn't really a part of my own research, but other work from my co-authors has given us a potential answer for this. And that answer is the presence of a native thistle species, Cirsium altissimum, or tall thistle. And tall thistle is closely related to bull thistle. It's a common species in many states and central United States. And it turns out that tall thistle hosts a community of native insects that are well adapted for feeding on the bull thistle. So when the bull thistle arrived in these communities where tall thistle was also abundant, it wasn't met with a suite of naive native species, but instead there was a community of insects that was ready to feast on it. And in fact, it was the bull thistle that was naive to the insects. So research has shown that these insects have a much stronger effect on the bull thistle than they have on the native tall thistle. And if we look at the distribution of bull thistle side by side with the distribution of tall thistle, we can see that in these states in the central US where bull thistle is not considered to be so invasive, they also have an abundance of these tall thistle populations. So it seems like the presence of this native species that is closely related to bull thistles builds up a community of native insects that is able to suppress the invasion of bull thistle. So finally, what are some of the big take-home messages for the management of invasive plants based on this research? So first, insect herbivores can reduce the abundance of invasive plants. And it actually seems like native insects might have a bigger impact than things like introduced biocontrol insects. So some of these biocontrol insects have actually been tried for bull thistle with little effect, in contrast to the huge effects that we saw of these native insects. And one thing that's likely really key to the success of these native insects is the presence of this native thistle that is closely related to bull thistle, because these insects essentially come pre-adapted to feed on the invasive thistle. And I'd argue that this biotic resistance provided by these native species is an underappreciated ecosystem service that they provide. So it's just one more reason that it's so important to maintain these communities of native species, because at least in some cases, they can help resist invasions, and preventing these invasions altogether is a much better approach than trying to control invasions that have already happened. All right, with that, I'd just like to thank some other people who were involved in supporting this project. So my graduate advisor again, as well as my dissertation committee that gave me feedback on this project along the way, and my graduate lab who just provided general support throughout this process. And then finally, my funding source when I was a graduate student working on this research, which came from the NSF Graduate Research Fellowship Program. And thanks to all of you for listening, and I'm happy to take any questions that you might have.
There we go, unmuting. Thank you, Emily. So for those of you watching online, please use Zoom's Q&A or the chat feature to, we already have a couple coming in, to submit any questions, and we'll have those relayed. So one that you've already touched on, Dr. Schultz, is that how common is it that native insects can control invasive plants? So that was just before your outro slide. And is it more likely that insect species would need to be imported to control invasives? And I had a very similar question for that is, what's involved with identifying an appropriate native herbivore?
So I don't actually know of any any efforts of trying to do that systematically. But I'm also more on the research side and less on the management side. So if anyone else in the audience has heard of anything like that, for sure, let me know. But I think, again, thinking about similar species and the environment, species that might be similar to those invasive species, might help us to determine if there are going to be any native species that might be able to control these species. So, having those adaptations that allow them to just get started controlling these invasive species is really important.
Another question is there's an observation that bull thistle is more prominent in areas where soil has been previously disturbed or removed. Is that kind of characteristic of bull thistles, and then to expand that further, invasives thriving in disturbed systems?
Yes, I would say that's very characteristic, both of bull thistle specifically and for invasive species in general. So bull thistle, I didn't show any data on seed production, but it can produce tens of thousands of seeds, a single plant. So they have these very rapid reproduction rates, which is one thing that can make them really good at resisting disturbance. Is, even if some of the species get killed off, they're producing thousands of seeds that can come back in and repopulate these areas. So things like being good dispersers and having high reproductive rates, which help species be good invaders, also make them really good at returning after disturbance like this.
And follow up to that, Kathy asks if there's any known insects that could be used to control cheatgrass. And I'll add to that Russian olive. Have you heard of anything in the research world that has explored biological control for either of those two invasives?
I haven't heard of anything specifically for those two. I've heard more about kind of mechanical removal and herbicide treatment for those two. I think often those might be tried first because there is a certain amount of risk associated with introducing a control agent. If it's another non-native species, there's always a bit of a risk that it could become invasive itself. So I haven't heard anything specifically about those.
So one of the questions that I had for you is that in your background, you've worked on invasives that occupy each of the different trophic levels, the snakes in Guam, the invasive herbivores on the Channel Islands, and bull thistle in Nebraska. Would you take a moment to speak to some of the similarities and differences in approaching both the monitoring and then the management at these different kind of trophic levels? Does that differ?
Yeah, so I'd say one of the biggest differences is how easy it is to detect and manage some of these invasions. So on the Channel Islands, the big invasive species there were, first of all, intentionally introduced. So these were things like sheep and cows and deer and elk that were introduced for hunting and for ranching. And they're also, very obvious on that island. So, there's no question that we have these invasive species on the island. They're very easy to find and monitor, and they're also much easier to remove. So that was one really cool thing about working on the Channel Islands is they have actually removed all of these large herbivores from most of the Channel Islands. And I was there at the time when they finished the removal on Santa Rosa. So it was much easier to get rid of those species than for things like the brown tree snake or especially plants. So the brown tree snake is maybe kind of an intermediate. There's a lot of trapping that goes on for those. One of the latest management strategies that I heard about was Tylenol-laced mice that they were dropping all over the islands because the Tylenol is toxic to the snakes. So there's certainly different types of management strategies depending on the type of invasive species and how easy it is to find it and get rid of it.
Like you have an extensive background in assessing ecosystem and species impacts of invasive species. What drew you specifically to look at biological control as a potential solution to the growing invasive plant problem?
Well, it wasn't really my idea on this project in particular. So my co-authors kind of came up with that idea, and Dr. Luda was actually working in that system for a long time. So most of the credit goes to her for identifying the insects in Nebraska as being potential controls for this. So I've actually done a lot of work on different types of control, like seeding with native species as well. So I think the thing that actually put Dr. Luda on this path to looking at insects was the presence of that native thistle, and they compared the insect populations on this native thistle and the invasive thistle and found that they were similar, and some of them were thistle specialists. So they were kind of, pre-adapted, specialized to feeding on this invasive thistle. So I think that's one thing that kind of led them down that path.
Yeah, I think that's a really good follow-up question. I think this is something we encounter with a lot of different things, but when it comes to these kind of management techniques and using insects, for example, are there any tips or advice that you would give on how would you go about changing or influencing people's perceptions at that negative perceptions of insects?
Mm-hmm. I mean, I think storytelling is really a powerful tool for helping change perceptions like this. So sharing these stories that we hear about less often about the positive effects of insects on these ecosystems. I think, often we hear about the biocontrol gone wrong and the insects taking over and being really destructive. So sharing these other stories about the way they make our ecosystem so much healthier and resistant to things like invasions, I think can be really, really effective. But it is a challenge. A lot of people just don't like having insects around, unfortunately.
Absolutely. Yeah. And we see that too with like the black-footed ferret and the prairie dogs. There's a lot of interest in black-footed ferrets, but there's not a lot of that same kind of emotional support, if you will, for prairie dogs. And so it's kind of hard to have one without the other there.
Right.
Go ahead.
I was gonna say, as someone who studies plants, I definitely relate to that. It's much harder to generate excitement about plants.
Mm-hmm. Is there anything else that you would like to share or would like our online audience to know?
I don't think so.
Well, Dr. Schultz, thank you for your time, taking time to visit with the Draper Natural History Museum and to share with us your expertise on the ecology and suppression of invasive species. Also, a big thank you to all of you, our sponsors, and to all of you in our audience here for supporting the Lunchtime Expedition speaker series. The presentation will be uploaded to YouTube before the end of the week here. So if you missed anything, I noticed there was a little bit of an issue with the registration link. So for those of you, any of you that have missed a first chunk of this presentation, that will be available online. So you can revisit as well anything that Dr. Schultz mentioned or discussed, you'll be able to find there. So thank you for tuning in. We hope to see everyone again on April 1st for our next lunchtime talk. Dr. Schultz, it's been a pleasure speaking with you. We wish you well as you wrap up the rest of this semester teaching at Northwest College.
Thank you so much. There is also
research. All right, one more question. Any current research folks want to know about here?
So I haven't done invasive species work in the last few years, actually. So most of my current research is focused on trying to understand tree distributions. So trying to understand if it's things like climate or competition from other species or disturbances like fire that determine where we see the range limits for species. And that can hopefully help us make predictions about how species ranges might shift because of things like climate change. I've worked primarily with both pinyon pine and white bark pine on trying to answer some of those questions.
Yeah, we should get you in touch with Dr. Diana Tombeck. She was a recent lunchtime speaker and is also working on white bark pine. So
yeah, I saw her talk. I have cited so much of her research, so it was exciting to get to hear her speak and ask her a question.
Oh, fantastic. Well, thank you again. For any of you out there in the audience, please feel free to reach out. The email there to sign up for any future lunchtime expedition talks. So please reach out, as well as with future suggestions as well. We're always open to those. So with that said, we'll see everyone again in April. So thank you again, Emily.
Thank you.